Welcome to Sci-ROI India's launch event, a virtual event for young Indian STEM professionals all over the world. It's a platform to facilitate the successful launch of early career STEM professionals in India by offering mentorship, collaborative opportunities, and scientific discourse. Now we are having the science showcase for early career STEM professionals in India. In this breakout room, we are going to have 4 talks related to structural biology and neuroscience. I'm Atul Gopal, a neuroscience postdoc at the National Institutes of Health, currently stationed in Washington, D.C. I will be moderating this breakout room. Each speaker will have a maximum of 30 minutes, a 15-minute scientific talk followed by a brief segment giving us a glimpse into their struggles as early career investigators. This will be followed by a 10-minute Q&A session. Attendees can unmute themselves and ask your questions during the Q&A session. You can also write your questions via chat, which I will read out to the speakers. We will try to accommodate as many questions as possible. I'll ensure that we don't encroach into the next time slot by keeping 30 minutes as a hard deadline for each speaker. The extra questions can be dealt with at the end of the 4th talk. With this, let me introduce our first speaker, Dr. Ashutosh Srivastava, Assistant Professor, Biological Engineering, IIT Gandhinagar. He is a computational biologist who uses modeling methods to understand the structure and dynamics of macromolecular complexes involved in circadian regulations. Let's welcome Dr. Srivastava.
Thank you so much, Atul. I'll just share my screen.
Sure.
Do let me know if you're able to see my screen.
Yeah, looks good.
Great. All right. Thank you so much, Atul, for that introduction, and thank you so much for giving me this opportunity. I'd like to thank Sci-ROI for giving me this opportunity to present some of the work that I've done in past, and if time allows, some of the work that we've been doing in our group for the last 1 year or so. So I'm Ashutosh. I recently joined about a year ago. I joined IIT Gandhinagar as assistant professor, and as Atul mentioned, I'm a computational biologist. So just a second. All right. So before we go ahead, I just wanted to give a quick glimpse of our beautiful campus here at IIT Gandhinagar. This is the bird's eye view of the campus along the banks of Sabarmati River. This is a housing block, Lal Minar, in an iconic kind of a building within the campus. And this here is an open air theater, which has a capacity of about 2,000 and several events that happen here. All right, we are a small group right now, just 6 of us in the computational biology group here at IIT Gandhinagar. 3 PhD students, Anthony, Shashank, and Shriyansh, postdoc Vidya, whose work I might be able to speak about a little bit at the end of the talk, and Rakesh, a master's student, and myself. We are funded by Ramalinga Swamy Reentry Fellowship, startup research run by SERB, DST, Inspire, CSIR. Before I go ahead and give my talk, I just wanted to thank all the people who have helped me throughout last 5.5 years, 6 years or so. So my postdoc lab at ITBM Nagoya University, Florence Stama's group. She also has a group at Riken, so I would like to thank my colleagues from there. And of course, my collaborators. As you will see, almost all of my work happens in collaboration, and I've been fortunate to have really good collaborations over the last few years. And so I would like to thank Tsuyoshi Hirota, Simon, Carrie, and this is the work that I'll be primarily presenting today, but also Keiko and Dushan. All right. So before we go ahead and discuss some of the work that I have done, I just wanted to give a glimpse or an overview of what kind of different studies that are done both experimentally as well as computationally when we are trying to study protein dynamics. And I saw Ati here, who's an expert in this area, of course. So basically, if we are talking about femtosecond to picosecond level dynamics of protein, which could be bond vibration, methylrotation, rotation, etc. Experimentally, we can use methods such as serial femtosecond crystallography, where we can visualize the reactions when they are happening. Computationally, we can of course use quantum mechanical simulations to study that. When we go to a slightly longer time scales to study the dynamics, where we would want to study, for example, side chain torsions, domain motions, etc. NMR spectroscopy is one of the best techniques that we can use to study all of that. At microsecond time scale, we can use hydrogen exchange mass spectrometry, again an excellent technique to study the protein dynamics. Computationally speaking, molecular dynamics simulation is one of the most common computational methods, actually encompasses this entire spectrum of timescales that we can study. And now even with advanced simulations that are happening, we can also go up to seconds or so. However, even now for studying for studying systems or for studying phenomena which happen at the time scale of seconds or so, we have to rely on coarse-grained simulations. On the y-axis here is kind of a size that I've displayed here, so usually for larger systems, we would want to go with coarse-grained simulations, and for smaller systems, all-atom simulations are fine. All right. So as I've mentioned, there are multiple techniques that exist if you want to study protein structure or dynamics. Each technique comes with its own advantages as well as limitations. For example, X-ray crystallography is an excellent technique for studying atomic resolution structures of proteins or other macromolecules. However, we do have issues, for example, missing residues. We have some radiation damage to crystals. It's difficult to understand post-translational modifications. Similarly, other techniques such as NMR spectroscopy, SACS, or cryo-electron microscopy, almost all the structural techniques that we have do have some or the other advantages or limitations associated with them. The computational methods which I've mentioned here in these green bubbles do try to mitigate some of these problems that are there, or some of the limitations that exist in experimental techniques. However, as we all know, there are limitations that exist within them too. So integrative modeling is essentially nothing but combining of data, or basically it can also be called as a hybrid method, where you combine data from multiple different techniques, both experimental as well as computational, to infer the structural model, the structure as well as dynamics of the macromolecular complexes. So having said that, I'll now move on to the system that I have been interested in for past 5 years or so, and that is the circadian rhythm, or the regulation of circadian rhythm, to be more precise. So this is a very simplistic picture taken from Wikipedia, which just shows that the physiology and the behavior of not just human, but almost all organisms on Earth actually varies as the day goes by. So it is synchronized in some way to the solar day. So we, for example, have fastest reaction time sometime in afternoon, which is actually hard for me to believe. We have lowest body temperature during morning. We have sharpest rise in blood pressure during morning. So all of these changes happen throughout our body, in human and in other organisms as well. In case of human, for example, these changes, or the perceiving of light, or the solar day, and then essentially the central regulation happens through a set of neurons, which are called suprachiasmatic nucleus, found near hypothalamus here. However, each and every cell also has its own mechanism to control this, or to regulate the circadian rhythm. Now this happens through a mechanism which can be referred as transcription translation feedback loop, and I'll just explain it in very simple terms here. Although it is much more complicated, there are multiple loops that exist here. So for example, here these two transcription factors, clock and BMAL1, they bind to a specific region of DNA called EBOX, and they lead to the transcription and translation of the proteins cryptochrome and period. These proteins in turn form complexes with other proteins, including kinases. These complexes then move inside nucleus, and then they go and repress their own transcription, leading to this negative feedback loop and rhythmic expression of genes. Now, why is this important? Because it has been found that any perturbation in these in the regulation of circadian rhythm can actually lead to a lot of diseases, sleep disorders, of course, but also cancer, cardiovascular, and metabolic disorders. All right, so where does our interest in this come in? So we have been interested in this protein called cryptochromes. As I mentioned, these are transcriptional repressors. They go and bind to the transcriptional factor clock and BMAL, and then they repress their own as well as other transcription. So these are very interesting proteins. They have evolved from photolyases, which are enzymes which take part in repair of DNA damage after UV exposure. And across different organisms, one part of this molecule of cryptochrome has remained more or less conserved in terms of sequence as well as structure. And there is another part to this particular molecule, which is a C-terminal domain or C-terminal tail, which shows considerable variation across different species. Now, coming to the mammals, there are basically two isoforms of these molecules or cryptochromes that exist, Tri1 and Tri2. And again, as I mentioned earlier, the structured part or this photolyase homology region remains very conserved, and the structure as well as the sequence are highly similar in these isoforms. However, we do see considerable variation in the C-terminal tail, which is also disordered in case of mammals. Now, although there is some kind of a redundancy that is present in terms of these isoforms, they are very similar, but there are very crucial differences that do exist between these two. And there have been several experiments which have now proven that. So basically, if you look at the mouse which don't have Cry2 knockout mouse, they show longer circadian periods. So basically, if the period is about 24 hours, it is longer than that. And they also show, or Cry1 alone also shows stronger repression. As I mentioned, cryptochromes are transcriptional repressor. Now, in case of mice that do not have Cry1 or in which only Cry2 is present, they show a shorter circadian period and weak repression. There are other differences that do exist, functional differences, but my collaborator, Dr. Hirota, was more interested in their isoform specificity towards small molecules that he was designing, and they were able to design, in fact, molecules that show isoform selectivity in stabilizing these molecules. All right, so as I have already told you, that they show differential binding to clock BMAL in terms of their affinity, and this was shown in vitro by my collaborators group, where they found that the binding of Cry1 is actually much stronger than binding of Cry2 to clock BMAL. There was another aspect to it. There's another layer of complication here, which is that presence of this another molecule, which is again very important circadian protein, actually regulates or somehow changes the affinity of these molecules. So as you can see, binding of PER actually decreases the affinity of Cry1, whereas binding of PER here increases the affinity of Cry2. All right, so we wanted to understand why these differences exist, and so we looked at the structure. Of course, it was very similar, so we started looking at the dynamics of it. And what we could find was there were basically two regions in these proteins which did show a significant difference in terms of dynamics of these two molecules. So I'm going to focus on just this one region, which is also called secondary pocket. And why this was interesting is because this is the place where it goes and docks to the clock molecule. In an earlier study, in fact, another group had actually shown that around 7 mutations around this pocket, if you change them, so if you mutate cri2, 7 of these residues in cri2 like cri1, then it starts behaving like cri1 in vitro experiments. So I was more interested in 2 of these mutations which were lying on the loop, which I found was showing difference in the dynamics. So I mutated these 2 residues in that loop, and what we could see is that mutating just these 2 residues actually leads to difference in the dynamics. Sorry, difference in the dynamics of mutated as well as apo cri2. My collaborator also performed experiments on these double mutants and found that indeed just mutating these 2 residues leads to an increase, 4-fold increase in the affinity of this mutated cri2. So we further wanted to understand how all of this is working together, and so we performed docking and a lot of other computational analysis. And what simultaneously my collaborator was working to solve the structure of this entire complex. However, that was not possible, and they could only get 7.2 angstrom resolution cryo-EM map. So we combined the models that I had got using docking and also simulations. We combined that data as well as the data that they got from cryo-EM map, and then we could come up with a pseudo-atomic model of this entire complex. Now, when we got this, they also solved the structure of this particular protein cry1 with period molecule, and for cry2 and period the complex already existed. And when we started focusing on the interaction of this period with the pocket that we were focusing on, what we found that the presence of PER was affecting the loop in different way in these 2 molecules. In fact, what we found is that binding of period actually was ordering the loop, a particular loop which was rich in serine in case of cry1, whereas it was disordering or causing some kind of a disorder in the loop which was getting ordered in case of cry1. So then we came up with this that period essentially acts like a molecular equalizer in this interaction between cry1 and cry2. All right, so I'll just summarize quickly that we found that the differential flexibility of this loop and the surrounding residues actually lead to differential binding of triisoforms, and period regulates the binding by interacting with this loop. There are others, so I did not talk about the other pocket which has another loop which is very interesting, and we are now actually trying to explore that. I don't have time to go into it, but we are also now looking at the C-terminal disordered tail of this particular protein through by combining the NMR chemical shift data and restrained MD simulations. This work is being done by Vidya, who's a postdoc in my group currently. All right. So as has been told to me, I am actually, so this is not what I'm going to discuss today out of several topics that were given to us to discuss. I'm going to focus on collaborations. As you might have guessed, almost all of the work that I do happens in collaboration with experimental groups, computational groups. So I'm just going to talk a little bit about collaborations. And it's not, I'm not going to talk about the challenges. I'm just going to talk about what I feel should be there if we want people to collaborate with us. So the first thing, in my opinion, which is extremely important when we are working in collaboration is transparency. It has to be there with respect to authorship, with respect to contribution. The next important thing which actually comes from transparency is trust. So if you have to be very careful when you're going to start a collaboration, but once you've started the collaboration, there needs to be a level of trust in terms of the work that is being done, in terms of capabilities of both the groups or multiple groups, as it may be. The third important thing which I feel which often goes wrong is timeliness or communication that has to be there between the groups. I'm not saying that we should keep harassing the other person or they should keep harassing us. But I would like to just mention that there has to be a constant communication because that is how collaboration grows. I also feel it's important to have in mind some tangible outcomes of what this collaboration will lead to, whether it will lead to a generation of some interesting data, whether it's just exchange of ideas or interpretation of data. And finally, and most important thing, I feel collaboration is a teamwork, and best of the collaborations work when they're bottom up. So, for example, some of the best collaborations that I've had in my career, very short career as of now, have been when I, as a postdoc, have been talking to different groups, and then it has gone on to the PI and such. So I feel those are the best kind of collaborations which are bottom up, where students or the postdocs lead the collaboration. However, even if it is top down, so even if the PIs have talked and then the students are working on it, I feel the priority has always to be the benefit or the well-being of the student or the postdoc who's working on it. So if you feel that the collaboration is not bringing any benefit to the student, you might be getting a very good paper. But if it's not bringing any particular benefit for the student, then in my opinion, that collaboration is not a good collaboration. You have to keep that in mind always. And with those thoughts, I think, and about a few seconds left in my time, I would stop my presentation, and I'm happy to take any questions. Thank you so much.
Thank you, Dr. Ashutosh. Anybody have any questions?
Yeah, so this is Adi here. Hi,
Adi.
Nice to meet you here again.
Yeah.
Atul, thank you for sharing this session and taking it forward. Ashutosh, I have questions for you on the mutation that you made. Serine to no, it was the mutation. There was alanine to glycine mutation. The other one was something, right?
Yeah,
or the
this was serine to asparagine and alanine.
Yes, serine to asparagine. So, and what do you think is really happening that results in, of course, alanine to glycine seems obvious that your fluctuation is going to increase.
Yes.
But what is happening with the serine to the asparagine? Is it inducing some kind of specific structural, change that is resulting in this particular difference in dynamics? Or go ahead. Please.
So actually, I did spend quite a bit of time to try to understand if there is any structural effect which might come in here. And I actually could not see any specific structural effect. There are some, so during the dynamics, I did see that some of the interactions have changed, which we would expect because there is a change happening in the entire pocket around the residues. But specifically, the structure, I could not find that these 3 or 4 interactions have changed, and hence there is a higher flexibility that might be coming in.
But so that has to be the case, right? So because you are introducing a bulkier side chain, and exactly. I see that the dynamics it's not just localized, right? It's affecting many residues
exactly.
farther away from the structure.
So basically, so in this plot, if you see, what was more interesting was the changes which are happening around this pocket. Which were happening, and so the other part which shows this huge differences, those are actually, that is something that I have not talked about, the other part of the molecule which has about 20 amino acid long loop, so that does anything at any point. So but so as I said, so basically at least I could not see during the dynamics, I could not see a specific interaction which was getting changed or which was getting broken or made, which might be leading to it. Sorry, I cannot be
no, but what happens if you just do A61 to G or S64 to N? Would you be able to
I was inclined towards that. But Carrie's group with whom we were working, So already another group had worked on 7 mutations. So 7 mutations around this pocket, the ones that I'm showing here. And they had experimentally shown, no simulations, no structure involved. They had experimentally shown that in vitro and in vivo, primarily these were in vivo results, where the Cry2, these 7M essentially started behaving more like Cry1. Okay. So Carrie was coming in from this 7 mutation point of view. And when she found that, okay, 2 mutations are doing this, she was more than happy, and they created this mutation and they found that, okay, this is working. So I actually did not want her to create now a single mutant and then find out if that works or not.
I understand.
So, yeah.
Okay, great work, and I hope I hope to see many more for the next.
Yeah, so actually we are now moving past this and focusing more on the disordered part of the protein, as well as the loop, which has been perplexing us for quite some time now. So hopefully something will come up there. Yeah. All right. Thank you so much, Adil. Thank you.
Dr. Srivastava, we have one more question from Shubhashish. So the question is, what are the experiments that they tried for dynamics? And I think he's referring to.
Hi, Ashutosh. Sorry if I can.
Hi, Shubhashish. Yes. Please go ahead.
Yeah, nice talk. So, like when you were discussing with Athi, so he says that group tried some experimental experiments, right? So I'm just wondering what kind of experiments they're doing for the dynamics. Is it something like FCS or?
No, so basically they did not do in this work, they did not do. So this work got published in 2020. So this work got published about 1.5 years back in eLife. So in this particular work, they did not do any dynamic experiments essentially. Okay, so they have not done any dynamic experiments. What they did was they solved the extracrystalographic structure of Cry1 in complex with PER. They also tried to basically solve this entire complex. So Cry, PER, and clock BMAL, which is the entire complex. However, the clock BMAL and the Cry, imagine this is a cryptochrome and this is where the clock is coming. This part of the clock and BMAL is highly dynamic, actually. It's a DNA binding region there.
Yeah, what is the time scale of the dynamics? Sorry.
So that actually, I don't know, because this entire complex has not been solved at all as of now. So it's just one domain which comes and docks here and becomes rigid. That has been, so even that has not been solved. But there are some SAX experiments which have been done on just this domain. Okay. And they tried to solve the cryo-EM map, but the cryo-EM map was available. So all that came up after all the data analysis was this cryptochrome and this particular domain, which is what we use to actually try to match our computational results to that. So apart from that, later actually Carrie's group has done NMR experiments on this protein. So we are now using that data, the NMR data, with respect to.
Sorry, and again, like, what is the time scale of the NMR? Do you know anything about that?
So basically in NMR, they are again, so this is a huge protein, about 500 amino acids, 580 amino acids or so. So there they have not looked at the structured part, for which already the structure is known. Now they are focused on the disordered tail that I mentioned right in the beginning. So that is about 100 amino acid, and they just looked at the chemical shift changes that happen when it comes in contact with. This was a PNAS paper that came last year. And now we are using that data to basically in our simulations to gain more understanding of how this disordered tail behaves when it is in contact with the structured region or the PHR region.
Makes sense. Thank you. Nice job.
Thank you, Subhashish.
Thank you, Dr. Srivastava. I think with this we can conclude this slot. If you have more questions, we can basically take these questions after the end of the fourth speaker.
Thank you so much.
Thank you. Our next speaker is also a structural biologist, Dr. Subhashish Haldar, assistant professor from the Department of Biology at Ashoka University. He and his team developed a magnetic tweezer technology that allows measurement of multiple physical properties of a molecule simultaneously within a single experiment in real time. Let's hear what Dr. Haldar is doing with this impressive new tool to understand molecular chaperones. Dr. Haldar?
Great. Thank you so much for the nice introduction. So, okay. Okay, let me start something with that. When we are talking about collaboration, so what I try to do is that I try to explain the technology so in an easy way, so that you can think about how you can use that particular technology in your research, right? So, yeah, I will talk about covalent magnetic tweezer. So we believe that this is a new window to see complex biological phenomena. Okay, so if I move forward, so let me get my lasers. Okay, so our lab is mostly divided into sections. We are one section try to develop single molecule technologies, single molecule methodologies, while is the other section they're basically involved in the understanding molecular mechanism out of those instruments. So I will give two different example. One is that real-time covalent magnetic tweezer. So we basically put it as a development part, and then we try to understand the mechanics of complex biological chaperones out of this technology. Okay, so now if you ask me that, okay, Shubhashish, we basically know that there is several single molecule techniques available. So what's new in your technology? Why we should use that? Straightforward question. So I would say that there is some unique advantage in this process. So where you can do a single experiment, again, mind a single experiment can provide you the properties of kinetics, unfolding, refolding kinetics, both thermodynamics. It can give you the information about conformational change. It can give you information about mechanical strength. You can give example of the polymer elasticity, right? So all these properties you can get with a single experiment. So not only that, so we can also see the real-time perturbation of all these parameters. Like mostly you can monitor all those properties. And if you want to see that how these properties are changing when you are adding protein or drug or any external solution, how the properties are changing. Very simple. One example is that maybe you are interested in protein drug interaction. So you can check that how the protein, all these parameters are behaving when you don't have the DNA, and when you add the DNA, how the kinetics or thermodynamics of the protein is changing. So you can do it with the DNA, you can do it with the drug, you can do it with the salt, with anything, right? So you can see all the parameters in real time. Now, if I say that, what is the resolution? I would say that our resolution is not only single molecule, we can detect up to a level of single bond, but with the time limitation, I cannot explain the single bond today, but if you are interested, I can talk about it. So we can even detect a direct bond breaking or bond making. Okay, now the other part is that, okay, so now the second question is that, okay, how much I need to spend to build such a technology? So, we basically build all these things in-house in Ashoka University, and that cost me around 3.5 lakhs. Okay, if you are thinking that how that could be so cheap, because I buy all the parts in eBay in second hand, right? So what is there in this magnetic tweezer? We have a voice coil. What is voice coil? It actually controls the position of the magnet. We have a magnet. We have a fluid chamber, which is just below the magnet. And we have CCD camera for taking some pictures, right? So whole cost of this instrument is 3.5 lakhs. Of course, I buy this microscope secondhand, but you don't need actually a microscope. You can do it without that as well. We only use the cage of the microscope. That's the whole point, and that's why I buy it in a secondhand. Okay, so how this thing works? So now you have to look at me, except on my slide. So you can see that I have a protein, which is my this end. This is stitched to a glass, which you can see there is a glass there. And at the top of that, there is a magnet, right? You are doing like that. So if you see my cursor here, so this is the glass, which is my cursor right now. This is attached to the protein with a halotech chemistry. We don't need to know what is halotech chemistry, but with some chemistry, I stitch the protein. And then at the top of the protein, I have a magnetic bit, right? So here I use 8 protein repeats instead of a 1 protein. That means 1 protein is stitched to another protein by repeats. And the point is that if you want, you don't need to be 8 repeats. You can do experiment with a single protein only, right? Okay. So this is how this works. So so where my cursor is right now, this is the fluid chamber. So it's a fluid chamber where all the proteins are, and this is the movement of the magnet, right? So when you are putting the magnet closer, the force increased, and when you put the magnet up, force decreased. It actually goes with the magnetic property which we learned in class 12 or class 10, and how the force is changing along with the distance. We basically published in before 2016 in JAX. Okay, so now I would like to show you a real data of magnetic tweezer, how it looks like. And when I said that this can calculate all the properties in a single shot, let me show you how it can do that. So it's a very complicated data of the magnetic tweezer. So I asked you to go very slow here. So look at this one. So here what we did, we basically changed the force. We go to 3 piconewton to 49 piconewton, and then come back to 6 piconewton, right? Hold it long and then go for 49 piconewton. Okay, again, this is important. 3 piconewton, 49, come back to 6, and then goes to 6 piconewton, 49 piconewton. So what do we see? When we go from 3 to 49 piconewton, if we see the length change, this is happening something like this. If you right-hand side can follow the thing, is basically one domain unfolded, and that gets the length change. So this is the whole point. So it will unfold one by one. This is very stochastic, and this is very common for single molecule techniques, that although we have the same protein, but they don't unfold at the same time. So here, if you basically analyze this curve, you can get the unfolding kinetics. Now, if I come back to the next place, where I pull the magnet up, if I pull the magnet up, the force decreased. And if you can decrease the force, you can see all the refolding of the protein, and now you can see the refolding. So now you can actually plot it, and you can get the refolding kinetics. And now you see, I keep it for 6 piconewton. I can keep it here for hours or even days. Like our highest measurement is for 14 days with one single protein. So if you keep there, you can see that the protein is actually hopping between the folded and unfolded state. So you get an equilibrium where you can calculate the delta G. Now the question is that why I choose 49 or 6 piconewton? Because I wanted to show you a good data. If I change the force like 10 piconewton, it will be unfolded slower. Or if I use this here 100 piconewton, it will be unfolded faster. Why 6 piconewton? Because here I can see you all the 8 domain refolded. If I use 9 piconewton, maybe there is one protein which will be refolded, right? So that's the whole part I wanted to tell. So we can also discuss more if you have any questions. So again, I have 8 domain, 8 repeats of a protein. So I have 8 steps. I have again 8 steps here during the refolding, and during the hopping, it basically hop between different folded and unfolded state. Interestingly, here you can see that I am basically going to state 7, because right, it is 8 domain folded. Now 7 domain folded. If I unfold from here, I should get 7 steps, not the 8 steps. This is how it is mathematically correlated, right? So from here you can calculate folding probability, and from the folding probability you can calculate the delta G. So as I promised, from a single experiment can give you unfolding kinetics, refolding kinetics, delta G, and of course the other parameters. Now this is basically where you get how these unfolding and refolding kinetics and equilibrium are. Now what you can do with this one, when we add the microfluidic system, we basically change the solution in between. Now this is again a complicated one, but let me explain you slowly. So where my cursor is right now, I went from 3 to 50 piconewton. Here is the force bar, you can see here. So 3 to 50 piconewton, you see that unfolding happens. You cannot see it here, but when I will zoom it, you can see that. Then I come back at 10 piconewton, you see only one domain refolded. So, and then I unfolded at 50 piconewton, I see one domain unfolded. Then, we basically introduce a salt, high concentration, which is 500 millimolar, and then refolded again. Now you see that at the same protein, right? Same protein refolded much, much more than what you observed so far. And then I unfold it again, I refold it again, put it at 4 piconewton and go to the 50 piconewton once again. Right, so there is a lot of complicated things going on. I don't know who missed what, but we can discuss all the topics during the question and answer session. So now the point is that if we see these two traces, if we combine that, we can exactly see that how much the unfolding kinetics changes with the single protein, right? So when we did that, we actually found out that the black one is the unfolding of the stress, which is very fast. The red one is the unfolding of the stress, which is slow. So, you don't need to do any calculation. From by eye, you can see that the unfolding kinetics is slower in presence of salt. So, right. So then you can actually see that step size different, that although you are putting the same force, due to the polymer size, it should have the same step, but it is less decreased. But I will not talk about the decrease in step size today because that's another topic. If you check the unfolding kinetics, again, you can see that there is a lot of refolding in presence of the salt. So you can calculate the refolding kinetics and the delta G, which I did here. So now I wanted to tell you that if I do the experiments many times, you can see that the data looks like this with the salt concentrations. The pink one is the 500 millimolar, and black one is the less salt concentration. And this is the curves are with the many salt concentration. But when we did the biochemistry on a single molecule, it just overlapped with the data which we get. Right, this is very nice. I told you that I will not discuss a lot about the decrease in the step size, but when we see the step size decrease is consistent at all the forces, when we did now the multiple experiment. So we would like to propose that, this is a biochemistry, but biochemistry on a same molecule. And then we showed that, when you do the biochemistry on the same molecule, whatever the result you get is actually comparable to the result of many molecules together. So, okay, now if you want to be a little bit fancy, what you can do is that you can actually apply the force like a force ramp, not in a force clamp. So what is force ramp means is that force clamp means you are increasing the force like stepwise. Force ramp means it is a constant increase in the force, constant decrease in the force. So you can also do that, and you are getting the same H steps. Good. Okay. Is there something going on? Okay. So, so now if we talk about the molecular chaperone, so so we all know molecular chaperone, that's actually help the protein to fold. So we have several molecular chaperones. So when the protein is coming out of the ribosome, it actually interacted with trigger factor. If it trigger factor can fold it, that is fine. Otherwise, it shifts the protein to the DNA system. If it can fold, that is fine. Otherwise, it gives the protein to GLOEL. Other set of protein with the help of chaperone, with the SEC-B and others, they are basically transferred to the edge of the cytoplasm, and they basically then pass through to the tunnel. And on the edge of the tunnel, there is another chaperone, DSB, which actually hold it. So now we try to address that, how the chaperone behaves when the substrate protein is under force. So far, chaperone has been known in such way that they are classified as holders and folders. So that holders chaperones are trigger factor DNAK, DNAJ. This is known for chaperone, and folders are also known. The interesting point is that when we apply force on the substrate protein, we saw that the result is different. Like trigger factor is not now holders, it work actually a strong folders. Now you can ask me that, okay, Shubhashish, what is the physiological relevance of working the chaperone with the force? So there is recent 2 papers shows that all the protein, they basically under the force of 1 to 20 piconewton, and in several biological process. And as we know, the chaperone, they are basically interacting with the protein with all the force ranges. So we like to know that how the chaperones behave here. So now you can see a real example that where the chaperone is working under force. I would only like to ask you to focus on this picture where the protein is coming out of the ribosome and it interacts with the trigger factor. And the paper Goldman et al. Shows that this force is around 9 to 10 piconewton when it interacts with the trigger factor. So till now, trigger factor is basically studied in absence of force. We try to understand how this basically helps the protein to fold. With the same protocol which we did before, what we did, we basically unfold the protein, and when we unfold the protein, we defold it at 9 piconewton, and then we found out that at 9 piconewton, only one domain folded. Here we can calculate all the delta G. And then when we add trigger factor on the same protein, you see that there is a lot of folding happens there, right? So on the same protein, we can see that at same force, there is a lot of folding happening, right? So here we can, if we plot the folding probability changes this way, but trigger factor is helping the protein to fold. Now I ask you to think a different way. remember that you are applying the force on the z-axis on the top, but the protein folds on the opposite side. So if the protein works against force, it is generating some mechanical energy. And here you can calculate exactly how much the trigger factor is generating the mechanical energy. Is there any chat from me? Because I cannot see it right now.
I think it's fine, Dr. Haldar. We can talk.
Okay.
We can deal with it after the talk. Yeah.
Yeah, because right now I can see in the
Yeah,
chat.
that's
Anyways.
fine. That's fine. Yeah.
Okay. So so the whole point is that trigger factor till now known as unfolders, but we found out that trigger factor is helping the protein to fold, so it works as folders. So we'll come to the point that what is the physiological relevance of that. But before that, we tried with a set of chaperones. We found out that DNA J, DNA K, or set B individually they are working as a unfolders. PDI, DNA JK, they work as a mechanical neutral, and DSBA and trigger factor, they are known as folders. Now, the difference is that DSBA and trigger factor in absence of force known as holders, but in presence of force, they are known as folders. I know this is difficult to remember, but just remember that trigger factor and DSBA, they behave differently in absence of force. Similarly, DNA JK is known as strong folders, and PDI also a strong folders, but now they are behaving as a mechanical neutral, right? So we did all the experiment with protein L. So protein L is a substrate protein, which is a beta sheet protein. So then we basically moved to another protein, which is taline. And now I would like to show that you always don't need 8 repeats. Here we did the experiment with the single domain, which is a taline protein, which is an alpha helical protein, and in vivo taline works under force. So very similar way as I did, as calculated all the values from the 8 domains, I calculated it for a single domain. It is a little bit easier. When I did the experiment, I found the same result, that DNA J, DNA K, they work as mechanical unfolders. JKE and PDI, they work as mechanical neutral. DSBA and trigger factor, they work as a mechanical folders. Now there is 2 important parts. One is that unfolded chaperone, they work as unfolders both in presence and absence of force. However, in presence of force, DNA JK is actually mechanically neutral. In absence of force, they are folders, and DSBA and trigger factor are mechanically folders, but in absence of force, they are mechanically, they are actually unfolders, right? So then we try to understand that, why these guys are behaving differently. What is the physiological reason behind that? So to understand the physiological reason, I'm trying to explain DSBA at first. So what DSBA is doing, DSBA is presenting in the periplasmic side, protein is presenting in the cytoplasmic side, and sec-A is the motor which actually translates the protein from cytoplasm to periplasm with the help of ATP. So one molecule of ATP translates around 20 amino acid of 20 amino acid from cytoplasm to the periplasm place. Now the point is that, of course, this can be worked well, but now we know that a lot of protein which don't have disulfide bond, they interact with DSBA. Okay, I forgot to tell you the story. The story is that DSBA is known as an enzyme which introduced the disulfide bond to the substrate protein, right? So now it is expected that DSBA should interact with the disulfide bonded protein, but we can see that there is a lot of protein which doesn't have disulfide but interact with the DSBA. So simple mechanism is that if it interact with the DSBA, and if DSBA helping the protein to fold, then this stops the backsliding, and this pole is basically people find it out, the force is around 9 piconewton in this pole, 9 to 10 piconewton during the translation. So if you basically find out that the protein is basically folded by the DSBA, so the extra mechanical fold, mechanical force will be generated to help the protein from translocated from the cytoplasm to the periplasmic side. Same thing is happening with the trigger factor, that when you have trigger factor, then trigger factor is helping the protein to fold on the translocon pole, which actually help in the translocation process. So very simply, we basically classify the chaperone in 3 ways in terms of force. We found out that, DNA K, DNA J, these chaperones, they work as a transferring chaperone, and due to the transfer, to stop misfolding or to stop aggregation, they basically slows down the folding, both in absence and presence of force. Mechanically neutral chaperone like DNA J, K, or PDI or thyrodoxine, they are strong folders in absence of force. but they don't work as a foldase in presence of force. The main reason is that these proteins don't need to work under force. That's why it doesn't show a foldase activity in presence of force. However, the tunnel-associated chaperone, both fear factor and DSBA, they are tunnel-associated chaperones. So these chaperones basically work as a strong foldase, and they basically fold the protein and which against the force. So that's all. I would like to thank the organizer for inviting me, and like thanks. This is my small group: Shorudeep, Madhu, Devajyoti, Deep, and Shoham. And this is our Ashoka University. You all are welcome to visit us. And these are the funding agency for the funding. And thank you. And I'm open to the question.
Thank you, Dr. Subhashish Haldar. Do we have any questions for Dr. Haldar?
Yeah, if it's fine, I would like to ask something.
Yeah, please go ahead.
Great talk, Subhashish. Very interesting. Single molecule experiments are already, are always very interesting to look at, and the kind of data that comes out is always mind-blowing at some levels. I just was wondering that when you pull this, and again, this is a very naive question, I'm sure. When you pull this protein and then relieve the force, how do you ensure that it actually folds properly, the refolding and the folding? So is this dependent on the system that this will always happen, or irrespective of system, it is system agnostic essentially that this refolding and folding will happen?
Thanks, Ashutosh. That's not a naive question. That's a very interesting question. So the whole point is that we have two options, actually many options. So one is that could be not properly folded. That could be goes to a misfolded state or something like that. To know that whether it is misfolded or that is properly folded, what we can do is that we can actually check the mechanical strength of the protein. So if it is misfolded, then mostly the mechanical strength decreases. So what we do is that after folding, after the folding step, which we are calling defolding, we unfold the protein and try to get the mechanical force, that at which force they are unfolding again. So we are checking the initial folded state and after pulse folded state, and we are comparing the mechanical strength. And when you get the mechanical strength, yeah, we can understand that whether it is folded or not.
Great. Another quick question, if the time is there.
Sure.
So across these different cycles, there's no, again, I'm trying to remember, hysteresis or something. There's no permanent change that does happen, after which you just can't do this experiment, or there's always this nice cyclical thing happens.
I only show you the nice ones. So the point is that when you are doing, I said that we can actually go for 14 days long experiment with a single protein, right? Yeah. So what happens is that at some point the protein loses elasticity, and then we cannot see the steps, right? Then we basically see the direct jump from the one to another. Yeah, of course. Yeah.
All right. Thank you so much, Subhashish.
Thank you.
A great talk. Yeah. Thank you.
Thank you.
Hi, Dr. Subhashish.
Hi.
It was a very nice talk, and I like the single molecule analysis of protein folding. I just wanted to ask you, you showed this schematic where you have the polymer, you made a multiple protein chain together. Is it really essential, you have to have multiple protein, or you can do it with one?
No. I guess, you missed the last part of my talk. So when I worked with Talin, I worked with the single protein. So let me show you the data once again. So, okay, let me show you. So
yes.
If you can see that, so the right-hand side is the panel where you can do with a single protein. So you see that at very low, at 8.5 piconewton, which is the below panel, it's mostly in the folded state. Try to jump to the unfolded state, but cannot. At 10 piconewton, little bit higher, you can see a 50-50 population of the folded and unfolded state. And from the little bit higher, like 11.5 piconewton, you can see that mostly it is unfolded state. So you have the whole energy landscape from the single protein with not a mark, right? And the same way, if you see here, you can also get the mechanical strength, here with the ramp. If you see that, yeah.
Nice, right. I just want to ask you about this. When you make this like multiple protein together, like actually how you are doing, what mechanism, how you are linking them, and when you are unfolding them by force, how you are differentiating whether it's one protein which is unfolding in multiple domains of a single protein are unfolding, and whether it's like different monomers of unfolding at different times.
Okay, makes sense. So this is kind of this slide, right? Yeah. So so there is 2 points. One is that how we are making the multi-domain. That's actually many techniques available in the literature. One is the golden gate technique. You can look for that. So you know, and the second point is that how do I know that single domain unfolding or one domain unfolding or one protein unfolding? So here we take the help of the polymer physics. So polymer physics says that if you have the whole protein, if you extend the protein, you can get up to a certain length. Beyond that, you cannot get that. So that's called the counter length of the protein. So the size of my protein is kind of 50 amino acids, so I should get a step size of 15 nanometer. But if it is a multi-step process, then maybe I should see 5 nanometer at first, 7 nanometer in the second, 3 nanometer in the third. So total would be 15, but it could be multiple steps. Also, the other point is that I took 8. Why 8? Because I want to use a fingerprint so that I know this is exactly my protein. So if I put 8 copy-pasted protein and if I see 8 steps and waited here long and still getting the 8 steps, that also indicate me that you basically get all the domains unfolded. And if the steps are matching with the counter length of the protein.
Yeah, great. Thanks.
Thank you.
This is Adi here.
Hi, Adi.
Shubhashish, I have a question on the trigger factor. Sure. I couldn't still figure out the force and the related aspects that you talked about. I'll need to think about it. But if you go to this trigger factor slide, the slide where you say, here, yes, this one. Yeah. So where you have this green arrow to the extreme left, you say that there is only one domain folded, right? So you cannot say that, right, really, because it can be a misfolded domain or misfolded conformation of a protein. That's possible because you have multiple domains that are linked together, and you could have a misfolding event that is really contributing to a state which is slightly, in between, if at all. So if you have specific steps while pulling, yes, I agree that you could, a pulling or where you actually move the domains apart, you can say that it's really coming from the unfolding of the protein. But when you're fixing the force at a certain point and then now you're letting the system relax, you could still have a significant degree of misfolding there with adjacent domains coming together, because distance is just one small coordinate that one looks into. And in the sense that it is not a good, collective variable really to know which regions of the protein are folded or unfolded. So how would you really say that it's misfolded or unfolded?
Got it. Yes. So that's a nice one. So yeah, I think this is basically a small trace which I showed you to show that, you can see only one domain refolded here and 6 domain refolded here. But the question coming whether it is misfolded or not. So what we usually do is that, we unfold the protein. We check that you can see this kind of trace, not exactly that step. Maybe I could come to this curve. So what you can do is that you are unfolding at a lamp. You see that how many domains are refolded, how many domains are unfolded. Yeah. Then here you can stay like long time, the refolding, not like that exactly here, but you can say that wait for refolding for a long time and then you unfolded it again. Right. So here you can get an average of the mechanical strength where the protein folded, and then if it misfolded, then the average mechanical strength decreased. You know what I mean? So,
but wouldn't it still be? You can always claim, or I know that's the simplest explanation one could give, but can you still say that it's not a misfolded state or a state which is like a bit different from what you would expect otherwise, an unfolded state?
No, there is a few other evidence as well. So, okay, so I don't have the data right now, right here. So, it's tough to explain. But the point is that so I have few things. So, if it is misfolded, then the step size which I am talking about, the step size could not be same as the folded state. It's actually different. This is our first judgment. The second judgment is that the mechanical strength is exactly same as that of the folded protein. So this is our kind of the second opinion to says that it's not a misfolded state.
Yes, but I'll just have one small comment on that. So the question which I still do not know whether it addresses the problem is the issue that the time resolution also matters. When you pull this, really what happens is that you might be seeing only those ramps or rather distance changes which are in the time range that you think are observable by this particular system that you are using. But if you have some conformational changes in the microsecond time scale, for example, would you still be able to capture them in this particular setup? No, right?
That's a good question. That's a good point. No, in a microsecond level, no, I cannot see anything in the microsecond level. Yes, that's true.
So what would be the time resolution for this experiment?
I would say 0.5 seconds, like 500 milliseconds.
500 milliseconds. Okay.
Yeah.
Okay. So anything faster you cannot observe is what you're saying.
No.
Right.
Yeah. But you can do other tricks, like you can slow up the force, so you can see a loose event. Like you don't need to go to directly to 4 piconewton to 50 piconewton. Instead, that you can allow the protein to 4 piconewton to 6 piconewton or 10 piconewton if you want to see some rare events.
Yeah. You
can detect on that. Yeah.
Okay. Yep. Thank you. Thank you, Shubhashish.
Thanks, sir.
Thank you, Dr. Halda. With this, we can move to our third speaker. Our next speaker is a neuroscientist, Dr. Mallika Chatterjee, assistant professor at the Amity University. She studies how the brain develops in utero, particularly the precise topographical organization of connections between thalamus and cortex. The proper connectivity between these structures is crucial for all forms of sensory and motor functions. Dr. Chatterjee?
Yes, hi.
Hi.
Should I start sharing my screen then?
Yes, please.
Yeah, okay. Can you see my screen?
Yes.
Okay.
You can move into the presenter mode.
Yeah. Where is it actually? Just give me 1 minute.
Yeah, you just need to press play.
Yeah, I'll just do that. Yeah. I was getting my. All right, cool. You can see it, right, in presenter mode now?
Yes.
Cool. Okay. Hi, good evening, everybody. A very late good evening. This is the latest I've ever given a talk. So thank you for the time, it's quite exciting. And since I'm more or less a late night person, so it works for me. And thank you very much, Sci-ROI, for giving me the platform to present my ongoing work, something which I've also carried forward from my PhD, and some ideas which we have developed in the lab with some collaborations, with some in silico work done with the help of some excellent colleagues in Amity University. And that's, you will see. So what I'll present here today is all about the brain. So the brain consists of billions of cells. But what is so unique about the brain is that it is a vast network of highways or nerve fiber tracks which connect the brain. Okay, so this is a typical highway network picture in Los Angeles, and now we have that in all major cities, both India and abroad. A lot of highway tracks, and you have a lot of cars going here and there, connecting all the major cities. The nerve fiber tracks that you see in the brain do the exact same thing. They connect the various cells and transmit information rapidly. So the billions of cells or neurons, as we call them, and also glia, by themselves, stand alone, are functionless. They only function when they connect and talk to each other. So these are the highways of the brain, also shown beautifully in this diffusion tensor imaging data, where you see all these complex fiber-like structures and coming out from the forebrain, from the cerebellum, from the spinal cord, etc. So they connect and talk to each other, which is vastly complex. So we cannot look into the entire complexity of the brain. It's taking us generations, it's taking us countries and intercontinental, like a human consortium, brain map projects, etc. So we can only look in our lifetime possibly into one major pathway of the brain. And in the starting of my PhD, I focused on one such important and very important highway that is called the thalamocortical pathway. The thalamocortical pathway is very important because it connects two key areas of the brain, the thalamus and the cortex. The thalamus is a key relay sensory station which basically connects information from various parts of the body and sends it upwards after some filtering of information to the upmost or the most, what we call the progressive part of our brain, that is the cortex. Therefore, the term thalamocortical pathway. We took our friendly mouse and used it as our lab model. And what you see here is a sagittal section, a section like this, of the mouse brain, which has been immunostained with markers. You see the thalamus here, it's a deeply located structure, nearer and closer to the other subcortical structures of the brain. And this has been marked green because it expresses GFP. And mind you, this is a transgenic mice, which expresses GFP under the promoter of a thalamic specific transcription factor called GBX2. And that I will use as a marker for the thalamus now. Our transgenic mice contained GFP as a marker for GBX2, a transcription factor, which is an exclusive marker of the developing thalamus. This is a coronal section of the developing thalamus, or rather the developing brain of the mouse at embryonic day 12.5. That means 12.5 days after it was conceived. And in the cross section, you can see this green area. These are the thalamic cells, that is the thalamic neurons. They later on will generate processes which will travel to the cortex. What we saw in this particular GBX2 mutant, which still continues to express GFP because it is the GFP gene is expressed under an IRES, was that these thalamocortical axons did not in fact go to the cortex. Where do they go? Many of them were stuck at an intermediate region in the basal ganglia, which is called the internal capsule. As you see, this is a higher magnification image. The F is a higher magnification image of this area in B. And we saw that many fibers, in fact, aberrantly crossed over, but they did not proceed all the way into the cortex. Even if they did, many of them had strewn and they strayed from the original path and they moved into the up into the file surface, which was very less if we noticed in the control. The most significant phenotype, however, or the defect which we noted in the GBX2 mutant was that as compared to the control, the thalamic cortical, now they are no longer thalamocortical in that sense, but these thalamic axons projected ventrally, that is into a region called the hypothalamus, which is again represented in the schematic here. This particular mutant phenotype or the defect of projecting into the hypothalamus was also observed by another group, Lopez Bendito, in 2007 in a series of mutants called ROBO1, ROBO2 mutants. What is ROBO? ROBO is a receptor for a particular chemorepellent ligand called SLITs of the SLIT family. So the SLIT family ligands bind to ROBO and initiates repulsion of those neurons which express ROBO family of receptors. So in the mice, there are two known receptors of ROBO, that is ROBO1 and ROBO2. What this particular group did was they placed a dye into the thalamus. In older days, it was called the dorsal thalamus, hence DTH. It's the same as the thalamus. DTH, in the DTH, a dye was placed, and this dye was tracking the thalamocortical axons. And what you see in golden or orange here is the thalamocortical axon, going out from the thalamus towards the neocortex. And in a higher magnification of the neocortex in the control, we can see that there are many such thalamocortical axons which have indeed successfully reached the neocortex. However, in the ROBO1, ROBO2 double mutant, what they saw was that the dorsal thalamic bunch of axons was no longer or very less in number, they were going to the neocortex. And so therefore you see a reduction in the intensity of the DTH labeled fibers. Instead, ventrally, when they noticed, a lot of these thalamic axons proceeded ventrally. This phenotype, as you can see, the schematic of the ROBO1, ROBO2 mutant as compared to the GBX2 mutant was somewhat similar. That is, many of these aberrant thalamic axons projected ventrally to the hypothalamus. So one of our questions was, since GBX2 is a transcription factor, does it act upstream of ROBO1, ROBO2 to regulate the expression of these two receptors? So that was the question we asked. And in fact, we saw that GBX2 does regulate the expression of ROBO1, ROBO2 differently in the developing thalamus. What we saw that ROBO2, this is an in situ hybridization experiment to detect mRNA transcripts of ROBO2 in the thalamus at early developmental stages like E12.5. And there we saw this is expression of ROBO2 in the thalamus. However, there is a complete absence of ROBO2 expression at the same stage in the GBX2 mutant. However, contrary to our expectations, we had expected both ROBO1 and ROBO2 expression to go down. What we instead saw was ROBO1 expression was not lost. In fact, it was ectopically expressed in those cells in the wild type thalamus. In the case of GBX2 mutant, it was expressed in those cells where it should not have been normally expressed. So basically, we could conclude that ROBO2 expression was downregulated and ROBO1 expression was upregulated. But if we remember the GBX2 phenotype, it seemed to recapitulate the ROBO phenotype. So it appeared based on this data and the previous, like phenotype characterization data, that even though ROBO2 was lost and ROBO1 was upregulated, ROBO1, which is known to rescue the function of ROBO2 in other systems, doesn't seem to rescue the function of ROBO2 in the GBX2 mutant, so as to cause a normal or revert to a normal phenotype of the TC normal condition of the normal status of the TC axons or the thalamocortical axons. So we came to the understanding or the hypothesis after doing a bit of literature search that it is possible that another third molecule called heparin sulfate proteoglycan might be doing this. How? ROBO is a receptor expressed on the neurons. It binds to SLIT, which is a ligand which is expressed in the surrounding area. And by binding of SLIT and ROBO, chemorepulsion of the thalamic neuron takes place. Therefore, in the wild type, they do not normally go towards the ventral side, that is the hypothalamus, and instead proceed towards the cortex because of the ROBO-SLIT interaction. However, if there is a third molecule called, as it has been in fact shown in literature, called HSPG, we'll just ignore this for a while, I'll come back to this. HSPG or heparin sulfate proteoglycan is a membrane-bound molecule which has a core protein and a sugar chain, which in fact does what? It binds to the ROBO and SLIT, this complex protein, and potentiates its binding further. That is, makes the binding affinity of SLIT and ROBO stronger. And this is something which has been already shown in the Drosophila and in other systems. However, it is not known what is the role of HSPG in the thalamocortical axon guidance mechanism. So we hypothesize that maybe in the GBX2 mutant, the HSPG formation or the function of HSPG was somehow compromised. Therefore, in spite of having equal good levels of ROBO expression, and in fact, we have also shown SLIT expression is unchanged in GBX2 mutant, both SLIT1 and SLIT2. Therefore, it might explain that why HSP in the GBX2 mutant, you do not have a proper ROBO-SLIT mediated repulsion away from the ventral hypothalamus. In order to find out we therefore then looked into the role of the sugars or heparin sulfate proteoglycans in thalamocortical axon guidance. And briefly, HSPG is consists of a core protein, as you can see here, which is binding to the plasma membrane, and to it is attached chains of sugar. And the sugar or the HS chain is not a simple chain of sugar. It consists of alternating residues of N-acetyl glucosamine and N-sulfated glucosamine. And the most interesting part is these chains may be selectively sulfated at various points. And the sulfation again takes place at various residues of these sugars, which is either at the 2-ortho position, which is a 2-ortho sulfate, or a 6-ortho sulfate. Okay, so that gives the potential of the HSPGs of having a lot of heterogeneity in its structures. And recently, in recent papers, it has also been shown that in interneuron diversity, this heterogeneity of HSPG has a very crucial role. So HSPGs are formed like this. There is first a polymerization reaction, then you have a deacetylase and an N-sulfotransferase reaction, epimerase to have glucuronic acid and iduronic acid for further modifications to the chain. And finally, as the last step of post-translational modification, you will have this sulfation happening at various points of the sugar residues. Because of this extreme diversity, the HSPGs are also well established, and more data is actually coming up on that they have a lot of diverse functions in potentiating already what is known to be existing molecule-molecule interactions. For example, slit and robo in our case. Okay. One example in developmental biology is also the fact that FGF, fibroblast growth factor ligands, bind to FGF receptors and help in a lot of developmental processes. It has been shown that HSPG co-receptors, in fact, again, potentiates their binding. Similarly, conformational role in conformational activation, enhancing protein-protein interaction, or helping to concentrate ligands like Wnt ligands, okay, on the surface of the cell for it to initiate an appropriate response, dose-specific response, has also been shown to be helped by the presence of HSPG. What we saw with the help of our microarray results in GBX2 knockout, that is the GBX2 mutant mice that I was just showing you, we did the microarray at the development stage of E12.5 because that's when the thalamus starts to develop. We looked at the thalamic gene expression profile from the microarray gene data list, and we found when we looked at the expression data for all the molecules that are involved in the HSPG post-translational modification, we saw significantly there was a downregulation of 3 isoforms of a particular enzyme called heparin sulfate 6-orthosulfate transferase isoforms. So what does this do? If you remember, I just mentioned that there was sulfation happening at specific positions in the sugar residues. Exactly 6-orthosulfotransferase enzymes, what they do is put the sulfation group in the HSPG molecule at the 6-ortho position. And in the GBX2 mutant, there seems to be a significant downregulation of the 3 isoforms that are expressed in the mouse. And we also have validated these results with qPCR and in situ. I'm showing you the example of an in situ result here, where sulfotransferase isoform 1, henceforth we will call it ST1 in my future slides, and ST2 seem to be significantly downregulated in the developing thalamus. We have also done ST3. I've not shown you the data because I have worked on mice mutants, henceforth the data which I'll show you on ST1 and ST2 double knockouts alone. Thereafter, we now looked into, okay, let's see what is the phenotype of ST1 and ST2, which is a short form of the enzymes HS6ST1 and 2. Okay. So this again is a coronal section of our mouse brain at developmental stage E14, embryonic day 14. And again, this GFP marks the thalamus. what we see here is considerable amount of green axons, that is thalamocortical axons going to the cortex. But in the mutant, they're in the double mutant, that is, they don't have the 2 isoforms of the enzyme. There seems to be almost no cortical axons going out from the thalamus. We did another experiment, which was an explant experiment. By explant, we mean like in vitro, we first take out the thalamic cells from the mouse, and then we culture them in presence of COS cells, which will secrete, depending on the control or the experimental condition, they will secrete either GFP or they will secrete the chemorepellent slit. So in the first panel, what you see here is a thalamic explant, which is wild type. It has just been given RFP for the sake of our visualization and labeling. And these green are the COS cells, which are expressing GFP, and they are grown in the same dish. The second one is the most important figure, where you see when COS cells start expressing the chemorepellent slit1, the wild type thalamic axons behave very strangely. That is, all the neurites or processes that you see growing normally in all directions. This is called proximal, this is called distal. This happens in the wild type, you get growth everywhere. But in the case where it is expressing slit1, what happens is all the neurites which were in the proximal end, that is towards the COS cells, are now moved, like they are repelled towards the other side. However, if you now compare this to the condition where slit1 is expressed in a double mutant, if you notice carefully, you will see the neurites or processes of the thalamic axon still growing towards the COS cells. And of course, this is the control of the decay with COS cells expressing GFP. So this comparison is the most important. And here it is apparent that the cells lose responsiveness or the chemorepulsive response to slit when they do not have the 2 enzymes, that is ST1 and ST2. And this is something which we show here also. That is, when we calculate the ratio of the proximal to distal dendrites, the processes, we can't call them dendrites here. There, what happens is, as compared to the wild type, where there is a clear loss in the proximal end of the processes, in the double mutant or the double knockout, there seems to be no response to the repulsiveness, or it doesn't repel at all. This is something which was also seen by us when we did mutant thalamic axons of GBX2. The same experiment with GBX2 mutants, they also lose the responsiveness to slit, and this has been already published by us. So this is similar. So ST1, ST2, lack of responsiveness to the slit-mediated chemorepulsion is similar in the GBX2 mutant. So since we are, neuroscientists, and we have very limited knowledge of, the biophysical aspects. So one way we are very interested in looking into what is happening at the molecular level, something which I am not yet able to attain because of my ignorance, because of lack of knowledge on how to do the experiment. So I approached bioinformatics colleague and biophysics colleague, who actually, Dr. Vijay Kumar, again from Amity University in my department. And therefore, we decided to do molecular dynamics simulation in order to see how was the interaction between slit, robo, and HSPG, and also in the mutant condition where you do not have the sulfated residues. So what we observed was when we looked at the energy subtypes, so in silico, what we observed after doing this docking and the simulation experiments, therefore, in fact, Dr. Vijay Kumar and his colleague Amrish also, he observed that there was a considerable reduction in all the different types of energy which was involved in their interaction in the case of the mutant complex. So the mutant complex, again, to remind you, slit, robo, and heparin sulfate minus the 6 orthosulfated residues. So there was an overall decrease in the energy of interaction of the components in the mutant complex. So Vijay also did RMSD, which was a root mean square deviation. And of course, I'm not the expert here. He would be a better person to describe it. But what I understand from at least a little bit of discussion with him is that there seems to be an overall increase in the flexibility of the three components, as is reflected in the, if you compare the red versus green height, so there is an overall increase in the flexibility of the slit robo mutant complex as compared to the control. And similarly, we see a similar change in when we also measure the radial gyri. And so this was further elaborated when we did over a 3D, like a conformational space. And therefore, the mutant complex seems to cover a larger area of conformational space than in the control. So we concluded that lack of 6-orthosulfation in the heparin sulfate moiety therefore causes the loss of stability, increased flexibility, and subsequently the loss of functions of the protein triad. To summarize my work, and something which I want to carry on as I'll share in my journey forward, is loss of ST1, ST2 partially phenocopies, not entirely, because some of the other mutant phenotypes we have not been yet been able to correlate it with the ST1, ST2. And because GBH2 is a transcription factor, there might be, it might be influencing other molecules, of course, which might contribute to its complex axonal guidance phenotype. But nevertheless, a part of it certainly phenocopies in that it doesn't reach the cortex. Okay, and again, a subpopulation of the TC axons are directed also towards the hypothalamus. There are some unique features that we observed in the ST1, ST2 DKO mutants, something which I am also trying to do the triple mutant in order to identify what if there's some redundancy because of the presence of ST3, is that axons are reduced in number towards the cortex, as I showed. Axon, again, some data which I didn't show for lack of time. Axon bundles from the pre-thalamus or region just beneath the thalamus are also shifted laterally. And finally, we have a sort of general model about what GBX2 was doing to this whole pathway of, or interactors, as GBX2 induces ROBO2 expression, it inhibits ROBO1 expression, it also induces ST1, 2, and 3. Together and individually, they are influencing the final interaction of RoboSlit and HSPG. So my journey so far is, I'll just show you some pictures of the favorite places and the mentoring which I have received from these places. I'm very grateful to all the mentors in my life so far. And I did my bachelor's in Presidency University. At that point, it was Presidency College in Kolkata, in physiology, from the physiology department. Then I decided to do Masters of Genetics from Science College, Kolkata. I moved on to US University of Connecticut Health Center, again, being trained by a great mentor. I'm ever grateful to my PhD thesis advisor, Professor Dr. James Y. Lee, for the extensive training and scientific background that he has created in developmental biology. For him, without him, I wouldn't be here today. And of course, great thanks to the lab models that I used at that time. We used mice and chick. I've done some work on chick also. Then I moved to the lab of Professor Shubha Tole in TIFR to work on hippocampus development and choroid plexus. Those papers are gradually coming out. And again, I worked on the mice. And finally, in 2018, I moved to Amity University, Noida, in the Amity Institute of Neuroscience, Neuropsychology and Neurosciences. And we have some great ideas which we have just initiated, and thanks to the generous support and guidance from various people in Amity University here, we are on our way to set up very vibrant research environment, because we are a new department. We've just started establishing ourselves. And so what is interesting is, in fact, we are setting up a zebrafish facility with the generous support of Amity for pursuing fundamental studies along with my studies, and designing therapeutics that are relevant to neurodevelopmental dysfunctions arising as a result of HSPG related molecular changes. At the same time, I am also a sort of struggling researcher, because I'm grant writing for core fundings, in the process of grant writing to address questions on, again, HSPG involvement. Especially, I have a special interest in autism spectrum disorder, because it has been shown that there are particular molecules with which HSPG interacts and whose dysfunction in interacting with HSPG might possibly contribute to autism. And we plan to use both mice and zebrafish in those studies. And again, a sincere thank you to Sci-ROI for allowing me to share my work and my ideas on this platform. Thank you.
Thank you, Dr. Chatterjee. Is there any questions?
Dr. Malika, very good talk, wonderful work, and very nice talk.
Thank you so much.
Really enjoyed it. So after seeing your talk, I have a question. When you are talking about this mutant mice, which have this bunch of neurons that moves towards the hypothalamus, I'm just wondering what is the behavioral outcome or the behavioral phenotype of this mice?
Interesting. That is something which we are aiming at doing, because that's something unfinished, and I plan to do that. And also, because you brought up this topic, HSPG is not only in the thalamocortical axon guidance, but also in other things like, as I think I mentioned, retinal guidance, axon guidance, and other various signaling pathways. So it would be interesting to just, with particular Cree lines or with optogenetics and all, like specifically do manipulation of ablation of those areas. Okay, just to see if particularly the HSPG in those areas is affecting the behavior relevant to the forebrain. Yeah, that would be an interesting topic to look into. Yeah.
All right, thank you. Because I was wondering, like, the visual cortex gets the bunch of information from the thalamus, basically.
Yeah.
So I was wondering, like, if this happens, then it might disrupt the functioning of the cortex, and as a result of which there will be, problems with vision.
Absolutely. Yeah. And we should look into the DLGN and all that for your connection also, thalamo-reticular, retinal, right?
Yeah. Thank you so much. Wonderful.
Thank
you. Is there any other questions?
Hi, Malika.
Yeah. Hi.
So this is Raja, and since you have kept me awake on a Friday night, I'm going to ask you a metaphysical question, which is, do you think that there is a heparan sulfate code as we know for the genetic code?
Well, I can't compare it to the genetic code right now, but it is already published. It is called the heparan sulfate code because there, as the variety that was created, I was saying, in the interneurons, it's been vastly studied that there is code for parvalbumin, somatostatin, and all that, which has specific signatures, like the type of sulfation that is happening is very different. So that core sulfation group is very important, how it is sulfated. So certainly it exists. It's not a metaphysical question. So the functions are not yet known. So it will be interesting to look into the carbohydrate, like the biochemistry behind it, certainly.
Yeah, thanks. We can talk more.
Sure. We'll catch up. Thank you.
Thank you, Dr. Malika. We can now move on to our fourth speaker. Our fourth speaker is Dr. Pinky Kane. Is also a neuroscientist. She's a principal investigator at the Regional Center for Biotechnology and a Wellcome Trust DBT Fellow. She studies gustatory processing, that is how our brain processes the sense of taste. Her research involves understanding how taste controls the neural circuits that regulates appetite and influences feeding behavior. Dr. Kane, you're muted. Dr. Kane, you are muted.
Can you hear me now?
Yes.
All right. And my screen is visible?
Yeah, it would be better if you could move into the presentation.
Yeah, just a minute. I'm just doing it again.
Yeah.
So is it visible now, full screen?
Yes, full screen.
All
But
right.
we are already in the second slide. If you, sounds good.
Yeah, I just moved to the first one. So thank you, Atul, for the kind introduction. I'm Dr. Pinkikin, and I'm currently at Regional Center for Biotechnology. My lab basically tries to understand taste and its modulation using Drosophila melanogaster as a model system. So I'm going to take you through our journey. Can you hear me?
Yes, we can.
Hello?
Yes, we
All
can
right.
hear you. Yes.
Because it showed me something on my screen. Okay, so I'm going to briefly give you the gist of the work that we are currently doing in the lab. So basically, there's a large chemical space around us in terms of various taste modalities. And the peripheral sensory input allows animals to accept food which are rich in calories and nutrient and reject food that are basically toxic or bitter in taste. And animals show attraction towards low salt, sweet compounds or umami taste, and they show aversive behavior towards high salt concentration, sucrose and bitter compounds. And generally, if we look at these responses are innate and largely invariant throughout animals' life, suggesting physiological hardwiring of taste quality to hedonic value in the brain. So how the taste detection start is through the peripheral organ, which is the tongue in case of human. But in case of Drosophila, which is a model of our choice, which you can see here, there are sensory neurons present on different taste organs. So these taste organs are the labellum, legs, genitalia, and the wing margins. And all these taste organs, they have small hair-like structures. We call them taste sensilla, where you can see these sensillae harbor or house gustatory neurons inside them. Apart from these are the internal organs called the pharyngeal organs, which are subdivided into three parts: labular sensory organs and ventral sebaral sensory organs and dorsal sebaral sensory organs. So all the information from the periphery, it goes to specific areas in the brain. It is called subesophageal zone, or in short, we call it SEZ. So if you look at the expression patterns here, the information from the periphery, which is terminating in the SEZ, is very stereotypic and basically based on from which organ it is coming from, and it is for which taste modalities. You can easily identify their patterns in non-overlapping areas. So one can, just looking at these expression pattern, one can easily say that, okay, this is from the leg or from labellum, or this is for the sweet or bitter. Just want to remind you that the neurons which are present here, they don't have sensilla-like structures. Basically, these are the internal organs where the neurons are present inside in the pharyngeal organ. These are like the portions we have in humans here. So there's a great deal of information which is known about peripheral coding of taste, both in humans and different animal models. But what is lacking is the understanding of taste circuits which are present inside the brain, where different parameters act on these cells. This could be the sensory information, different physiological state, and how the memories are formed, how these neurons are connected with the peripheral neuron that finally gives rise to the behavioral output. So in my lab, what we are trying to understand basically is trying to find out projection neurons, interneurons, motor neurons, or any sort of neuron, basically that talks with the peripheral neurons and translate these peripheral signals into meaningful behaviors like acceptance or the rejection of food. But apart from this, we also look at pharynx, labellum, and leg. So I'm going to give a gist of 2-3 different projects where we have looked at the pharynx and into the brain, looking at different circuits. So one can actually look at the periphery by doing the recordings, and they can actually do the electrical recordings by these sensory hairs which are present on the labellum or legs. The recordings from the pharynx is very difficult because they don't have hairy-like structures. They are internal organs. So to understand how the taste modulation happens in this place of the fly structure, we started looking at one of the parameter, which is salt or general table salt. Other things that can actually modulate the taste behavior is basically long-term use of drugs, some metabolic disorders or prolonged illness. There could be an effect of neurotransmitters or neuropeptides and various physiological states. So right now I'm just going to concentrate on salt. And why salt? So basically, we all know that humans, including other animals, which are shown here, we have an innate liking for salt, just like sugars. And the presence of salt in the food makes the food more palatable than without the salt. So it is shown here that, you know, when there's a requirement of sodium present in the salt, animals will start, you know, finding sources in their environment to lick the sodium from this, like the cow is licking the rock salt and the butterflies are puddling on the urine, and these crickets are cannibalizing each other, or the mosquito sucking the human human blood. I'm not sure if this is working. So what all this suggests that sodium is a very important ion that we need in the body, which is required for the nerve transmission, to prevent muscle cramps, for absorption of nutrients, and to maintain the electrolyte and fluid balance. And without sodium, like overconsumption of sodium actually can lead to many diseases which are mentioned here. So sodium present in salty food, basically we cannot ingest, we cannot make it by our body cannot generate sodium by itself. So we need to take it or ingest it from the external food sources. And when there is a low sodium in the body, it triggers an appetitive signal in the brain that drives the consumption of sodium. And what is happening right now in the Western world, that according to the WHO guidelines, which suggest that adult human beings should be taking only 1 gram of sodium per day, we have been consuming much more sodium in our diets because we are depending on the packed and fast food for our daily needs. So to understand the mechanism of high salt consumption in animals, we looked into the neural mechanism of high salt consumption in adult Drosophila, by which flies actually modify their acceptance of high salt as a function of diet. So generally, if you look at this graph in the gray scale, gray bars, they show that lower concentrations are attractive cues for the fly. This is for the salt. And as the concentration of the salt increases, flies show aversive behavior. But when these flies were put for 3 consecutive days on high salt diet, this shows a modulation of salt behavior, or it alters the salt behavior, which was preferred earlier. Now these flies start liking the higher concentration of salt. And basically, this is too high, you know, basically these concentrations are generally bitter for the fly. So what we have found that this kind of modulation actually requires pharyngeal neurons, the lamellar sensory or the alesopharyngeal neurons, where this kind of modulation takes place. So if we silence the peripheral neuron, it could be a bitter neuron, it could be a sweet neuron, or any category of neuron. If you silence the peripheral neuron, leaving the pharyngeal neurons intact, you see a huge response in feeding behavior. And if you silence any category of neurons at this level, so I'm talking about the pharyngeal LSO area, if you silence any one type of neuron, these responses that you see, they come back to normal level. So suggesting that different pathways and receptors are involved in this kind of modulation, and they act at the very first level of the pharynx in flies. So what our data suggests that both starvation and dopamine are important to cause such kind of modulation. And we have seen that these flies show high salt intake. Otherwise, in the fed state, we don't see such phenomena. So what we don't know at this stage is that this neuromodulation, is it only happening at the pharyngeal neurons, or it could also be happening in the taste circuits which are present in the brain. So we are looking at this, but what our data actually suggests that flies can adapt to the amount of salt ingested over several days, indicating the presence of a critical mechanism to reset the salt appetite and related neural circuits. So to identify circuits which could be involved in sensing appetitive cues like salt or sugar, we performed a screen in the lab. The expression patterns are shown here. So remember SEZ, which is a primary taste center in the brain. We found lots of neurons, putative set of neurons, which were present in and around SEZ. So we next asked which one of these are actually sensing the appetitive cues. So for this, instead of doing the feeding behaviors, we used temperature sensitive D-TRPA channels. We drive this channel into these neurons and asked if these neurons can be activated and at high temperature, and if they can extend their proboscis towards, if they're a part of an appetitive circuit, they will extend their proboscis. So we found that many of these neurons showed a response which was around, more than 50% or 45%, suggesting that these could be the potential subset of neurons which could be a part of appetitive circuits. So generally for these experiments, we put the flies on 34-degree heating plates and see if they are extending their proboscis, which you can see here that as soon as they are put on high temperature, they start extending the proboscis if these neurons are present, are sensing the cues. So instead of providing a sugar or low salt concentration to these flies, what we are doing is we are raising the temperature and asking if these are actually a part of appetitive circuit which are present in the brain. So one such circuit that I'm showing you here is this. So if you look at the cells in green here, it belongs to cluster 1, and these neurons make synaptic connections with the peripheral sweet taste neurons. So we used a technique called grasp. So basically we use different fragments of GFP here, and only if these fragments are coming together and shaking hands like this, you will see a signal here which suggests that these neurons are synaptic partners. So we found one subset of cells here, and then we wanted to know what are their physiological functions. So after doing the calcium recordings, we found that these neurons also respond to sugar cues or sweet cues, just like the peripheral sweet taste neurons. So if you look at their patterns, they show a beautiful increase in a dose response curve when the sugar concentrations were increased. And also these neurons show increased sensitivity towards sugars under starvation and dopamine. So just to hunt for other areas in the brain, we also found a novel area. It is called central complex area, which is shown here. So central complex is one of the regions which is, so this is SEZ, it is above the olfactory center. So what you are seeing here is the complete central complex area. It is subdivided into many parts or cells here. So we did another screen to see if there are any other regions in the brain which could be involved in regulating feeding behaviors. So we found that the ring structured neurons, if you silence these neurons, they also show defective feeding responses, which is observed by looking at how much flies are feeding in a plate assay or looking at the consumption of flies in absorbance assays. We also wanted to confirm these results. So we again did the PER assay or the proboscis extension assay. So these were done manually and by expressing D-TRPA, which I just showed you. So in both the cases, you can see that silencing these neurons brings their responses brings the responses of this set of neurons to a lower level compared to the control set of neurons. And the responses that we have observed for D-TRPA, it is just like the sweet taste receptor, suggesting that these neurons are indeed part of the sugar circuitry present in the brain. So the next question was to basically understand what are the neurotransmitters or neuropeptides that could regulate these behaviors via these neurons. So we expressed short neuropeptide F because it is a well-known neuropeptide which is responsible for central regulation of feeding intake in vertebrate during the vertebrate development. So what you can see here under fed state, if you knock down SNPF, you see reduction in feeding responses, but you overexpress. Upon overexpression, the feeding responses were increased, and the reverse was found under starve condition. Similar results were observed when we knocked down the receptor for SNPF using RNAi line or dominant negative form, and opposite was seen in the starvation condition. So this is very interesting because this is something that we are seeing under fed state, not in the starved state. And similar results were also observed when we wanted to check if dopamine signaling is playing any role in these set of neurons. And here also you can see that down-regulating dopamine 2 receptor causes increase in feeding responses, but the starvation state brings down the responses to a normal level. So we did not see any phenotype in the starved state. So one obvious question is, since the central complex is a conserved area, conserved brain area in many other insects, which helps in path finding and calculating the distance and integrating all the information in ants, bees, grasshopper, or cockroaches. So one obvious question that came to our mind is also to look at the foraging behavior of these flies. So what we did, again, we tested the flies after silencing the neural activity of these neurons using tetanus toxin. And in this case, just doing the general recording of the fly foraging, we could find that under the starve condition, these flies, they lose motivation to forage, even if they find the sugar drop in the center of the plate. In this case, you can see that this fly just walks away from the arena. So during the foraging behavior, we did not see any difference in fed state when the neural activity was silenced. Under starvation condition, they showed reduced foraging. So we did many other experiments using SNPF and dopamine and other molecules. As a whole, what we have understood from this story, I can't show you the whole story in 15 minutes, but what we have understood is that we have found a circuit which is a satiation state dependent circuit, meaning that whatever phenotypes we were seeing in the feeding behavior, they were all during under the fed state. So we have seen that the SNPF level or the knockdown of SNPF, PDFR or dopamine 2 receptors, which are present on the same set of neurons, are regulating feeding behavior or modulating feeding behavior under fed state. Since SNPF also works through insulin signaling, we are also checking if insulin signaling is involved here. Dopamine 2 receptor acts through G protein signaling. So we are also trying to see where they fall in the pathway and how these signalings are also, incorporated here and regulating the feeding behavior. But for the foraging behavior, not all the receptors gave us the phenotype. We just found that when the SNPF levels are very high, or if there is a knockdown of DOP2 receptor, we see increased foraging and feeding. So again, suggesting that it is a circuit which is basically fed state dependent or satiation state dependent. So these findings provide evidence for active foraging in well-fed flies that is separable from hunger-driven foraging. So generally, if you look at the literature, you will find neurons where you see alteration in the foraging behavior during the starvation state. So this is a report where first time we are showing that these are involved in the fed state and multiple receptors are present which are regulating both these kind of behaviors. So with this, what we are trying to understand in the lab, I could not tell you all the stories that are going on right now in the lab, but what we are trying to understand is apply all the results from the fly to other model system. In fact, we have started working on one other model system to save agricultural crops or, design insect traps, or maybe use it for mosquitoes, which are basically which generally feed on human blood for the reproduction and all, and also to see how the taste modulation is happening and if that could bring out some better taste modulators, and to understand metabolism and obesity or other food related disorders. So I hope that at some day we will have solution to all this. And with this, I would like to thank people who were involved in these projects, my funding, my collaborators, and various people who have contributed reagents or fly stock to us. We also try to take our science from the lab to the society. So we have been involved in many science outreach programs, and we teach kids about being, healthy and happy all the time, about mental health and how the eating habits actually can help them a lot for their well-being in general. So thank you so much, and I'll be happy to take questions now.
Thank you, Dr. Kane. Is there any questions?
Hello. Sir, am I audible?
Yeah, yes.
Hello, ma'am. It's really nice hearing you. Ma'am, my name is Aman and I'm from JNCSR. If I'm not wrong, I was a little confused between, you started with the NaCl sensitive neurons and then moved to the sweet taste neurons. Between there, you're talking about the heat sensitivity, temperature change, with respect to study the NaCl sensitive neurons, right? I was not able to fill the gap between the NaCl sensitive neurons to the sweet taste neurons.
Okay, so basically, I could not show you the video where we see extended proboscis in terms for low levels of salt like 10 or 50 millimolar. Okay, so when I'm showing you different circuits which are appetitive circuits, I'm calling them appetitive circuits because they could be recognizing sugars, they could be recognizing low salt or umami taste. So from these circuits, because we have done different feeding assays, none of them has worked for the salt. And that's why I said that what we have found so far are the sweet circuits. So the basic idea here is to show where the taste modulation could take place. So one place is pharyngeal area, and there is some data where people have shown, not in terms of pharynx, but they have shown that there is a peripheral modulation of sweet taste. So I did not touch upon that. We looked at the salt taste in our case, and the role of pharynx is not well known. That's why I thought, it's nice to show how pharynx are modulating, how the pharyngeal neurons are modulating the salt behavior. And then I moved on to circuits because, we were aiming to get some salt circuits which could be recognizing low or high level of salt, but we haven't really found anything so far. What we found were for the sweet taste, basically. So
okay,
I was just trying to give a gist of, different questions that we are trying to do it in the lab right now.
I got it, ma'am. Last question. So apart from the sweet taste neurons, what other tasting neurons we have in Drosophila? I want to know their taste appetite also.
Basically, Drosophila can identify any taste, So it could be sweet, sour, bitter, umami, water, carbonated, anything you ask for, Drosophila can differentiate any taste, fatty acid, fats, whatever it is, amino acids. The only thing is that slowly the field is evolving because there are gustatory receptors, there are inotropic receptors. The function of all the inotropic receptors is not known. Slowly people are trying to understand and finding the right ligand for them. And then for the gustatory receptors, 50% of the receptors are known to basically identify bitter ligands, but the ligands are not known. So the field is still unexplored to some extent, and we are trying to understand what are the receptors that are required to recognize other taste modalities.
Right, ma'am. Thank you so much. Thank you so much.
Dr. Kane, you mentioned about the effect of dopamine on these neurons.
Yes.
I assume that these are peripheral sensory neurons, right? Can you elaborate a little bit on how dopamine affects these?
Which one are the peripheral neurons?
The neurons that you were talking about were mostly...
Okay, so basically, in the pharynx and in the taste centers, which we have found in the brain, sweet taste centers, for example, in both the cases, what we have found that when you put the flies on under starvation, you see increase in sensitivity of the neurons. Okay, the same effect you see when you feed the flies with dopamine and you do not starve them.
Okay, so you can directly feed the flies dopamine.
Yes. So what we do is, instead of starving the flies, we feed the flies with the dopamine in the media for 3 days. And
Okay.
then we ask if you see an increase in the sensitivity of a particular compound like sugar or salt,
Okay. Okay. Thank you.
because it acts like a reward system for them.
Okay. Are there any more questions?
So I also have to talk about one of the topic, right?
Yeah, that would
So
be
I
great.
will just briefly say, I just wanted to say that, yes, as a women scientist, I also faced lots of challenges, during postdoc and even later. And one of the main challenge that I faced was during my pregnancy, because, it is very difficult to manage work and child at home, especially when you have nobody at home who can support you except you and your husband. So we had a very tough phase, and we were struggling every day. We were taking shifts to take care of the baby. But I think the one point is that you really have to decide what you like to do. So you have to show determination, you have to love your work, you have to love science, and moreover you have to organize things in a manner that you can balance family life and the professional life. And on that, if you have a supportive family, I think nobody supports you except your family, because they are seeing you day and night, and they wake up and sleep with you. So if you have a supportive family, I think that really helps. Even if they don't belong to scientific field, it is fine, but if they are supporting, it really helps. So I would just like to say that if somehow we learn to balance, and somehow if we learn to organize things well in advance, it really helps, because these are the challenges that we can't avoid. Everybody is facing that, especially women scientists, when it comes to delivery and all that, but we have to learn to deal with this.
I'm on the same line. I would just like to ask, how was your institution's support while you were going through these things? Did your institution supported you, or were there any mechanisms in
Actually,
place?
I was lucky that I didn't really face any challenges at that time, because when I joined RCB, things were being ordered, and that was the right time, fortunately for me. So I ordered lots of things in the very first month, and they were just delivered on time. And also because we are close to Delhi, I think that makes a big difference. So the good thing is that I already knew what all I need, because my Welcome Trust grant was there. Everything was charted out properly. And, I had students, I took the students in the very first month. So all that really helped. I really got a good support from RCB. Yeah.
Thank you.
They have a good central facility as well, which helped me a lot.
Right. Do we have any more questions? Thank you, ma'am, for joining us for this
Thank you, Atul.
session.
Thank you.
It was a wonderful talk. I look forward to hearing from you more. Thank you.